Hello! How y'all doing today? Alright, we're going to start in and jump in on, basically from here on in, we're going to start kind of chipping away at each part of the different kingdoms, although we're going to kind of start on the domains first. So we're going to look at two of the three domains today, which is Archaea and Bacteria. Remember, there's three domains. There's Archaea, Bacteria, and Eukaryota. And that's where the rest of the kingdoms come in. That's where when we hit the eukaryotes. So remember the differences between a prokaryote and a eukaryote. Prokaryotes are very small. They do not have membrane-bound organelles like eukaryotes do. Usually these things are Archaea and Bacteria. They used to be called Kingdom Monera, but we've dropped that because it's become much more intricate than that. So we're not really going to be using that terminology. Now, I said this I think I showed this a bit at the end of the virus. This is called The History of the Entire World, I guess. It's long. And it's mostly focusing on human history in a very humorous way. However, and I would like, honestly, it's pretty good, at least, and for the first, oh gosh, I want to say 10 minutes until it gets to humans again. That's basically what we're kind of going to be talking about, so I'm going to reference this a bit again and again. It's still one of the best funny representations, I think, of our current understanding of how everything came to be in science. And although there is a lot of swear words, there is a clean version out there, but I don't think it's as funny. But anyway, I highly recommend, if you haven't already seen this, it is worth a giggle. If you want to watch the whole thing, have fun. But like I said, the first about 10-ish minutes is basically what we care about, where it goes over, how we think life popped up on Earth. And actually, before that, it kind of starts at the Big Bang and all the fun physics behind that. And like I said, it's a humorous take. Highly recommend a watch. But like I said, if you're not a fan of swear words, you might want to skip it or find the clean version. Somebody made a clean version. But anyway, so yeah, this is The History of the Entire World, I guess, by Bill Wirtz. He also did a history of Japan, I guess, which is also just as amusing. And then after that, he didn't make much more, so I don't know what he's been up to. I haven't looked it up. But honestly, good videos, and it's just a bite-sized way to remember how things go. Even crazier space dust. Anyway, so Bacteria. So if you watch the video, he mentions microbial mats, or these large biofilms, and that's basically what happened, is down in the depths of the ocean, because the ocean was safe, relatively speaking, for life to form. Also, where did it come from? We're still trying to figure all that out. We've got, a lot of fossil evidence, and I'm going to talk about some fossil evidence in a minute, because I used to live on top of that fossil evidence when I lived up in New York. So we'll talk about the microbial mats in a minute. So anyway, the earliest form of prokaryotic life that finally got together and did this cool thing with DNA and RNA, and it could reproduce and split into two through binary fission and all the things we've talked about back in 111 and all that fun jazz. Yeah, so did it come from chemistry coming together and coming together and coming together because of energy usage and whatnot? Or, another thought is, here's a weird one, that a chunk of, because there might have been life on Mars at that point. Mars may have been a completely different planet than what we know today. And rock popped off from Mars and hit Earth, and we might all actually be descendants of Martian bugs. And by bugs, I mean bacteria. So there's that thought too. I mean, it gets interesting. So anyway, but there's these microbial mats. So basically bacteria figured out how to live together. So they decided to make these giant biofilms, and it's a massive microbial community held together by this gummy-like texture matrix that consists of polysaccharides secreted by organisms and some proteins and nucleic acids exchanged. Because bacteria does some weird junk, like they like to trade DNA loops which is the plasmids. They'll just be like, oh, I don't have that plasmid. Do you have that plasmid? And they'll switch out DNA plasmids between each other, like, Pokemon cards or Yu-Gi-Oh cards, or, the granddaddy of them all, Magic the Gathering, which is what I play. Anyway, so, and we actually have fossils of these guys, these microbial mats. So this microbial mat is a multi-layered sheet of prokaryotes. It includes mostly bacteria, but there's also some archaeans. Remember, archaeans are even more primitive than bacteria. And we believe, or at least we have evidence pointing to archaeans gave rise to bacteria, but they also hang out side by side. That happens quite a bit on this earth. A lot of people have this thought where it's like one exists and then they're gone and then. And in some places, yes. And in some cases, they're side by side. So we still have archaeans today. We still have bacteria today. So just because they're ancient doesn't necessarily mean they're gone. So, and they typically grow where there's basically where they've got their main things. And remember, we went over back in 111, biotic and abiotic. What are the abiotic things life needs? And one of the things is water, and how crazy important water is. So as long as there's water, that's where these microbial mats showed up, and they just went. And we do have fossil evidence of these guys. We have these stromatolites, which is these sedimentary structures formed when minerals participated out of the water and by prokaryotes in the microbial mat. And these are actual pictures from where I used to live up in New York, because literally there's entire swatches of the state of New York that's just, if you dig down, you hit this layer, because New York used to be a swamp. And as I like to call it, swamps where all the fun's at. And that's what these guys are. And again, this was right next to my old house. And this is called Leicester Park. They're 490 million years old, fossilized bottom of a seabed. And these guys are just insane. They're round gray shapes, stromatolites, or layered stones, which were basically these microbial mats. And their layers reflect growth spurts to tidal cycles, and many marine animals such as snails, trilobites, brachiopods have been found in between these guys. So again, these microbial bats have been hanging out forever, as long as everything was right, and other creatures found their way in living with them. So think of it like a gummy, icky, slimy coral reef that we have today. You go down underwater, go to a coral reef, and oh my God, there's all this life hanging out in between and everything, and it's a whole thing. So imagine a gummy version of this with just prokaryotes. So these guys are, right down here, if we look at all the different timelines. So we got the Precambrian, which is the origin of the mountains up in New York with the Adirondacks and the Hudson Highlands. Mountain building, oldest bacteria fossils, and then bam, the Cambrian. And that's basically where we're starting right here. So, and then up to the Ice Age. So anyway, so these microbial mats, they've been hanging out forever. It's some of the earliest stuff we've got of bacteria hanging out on Earth. And here's some random dude and my son running around in this. Sad part about this is it's right next to a giant open pit quarry, which they are constantly booming. But the crazy part is you're like, well, why don't they preserve? This is technically preserved. It's owned by the Natural History Museum of New York. But the reason they don't pull it up and put it somewhere else or something like that is they're worth more in the ground than they are somewhere else. So it's been a fight between the quarry on the other end of this hill and all these fossils. But it's a fun thing. I just wanted to show you that, yes, we do have fossil evidence of some of the earliest life on Earth. So if somebody sits there and goes, well, do you have evidence? Yes. Actually, in some parts of the United States, you can just dig down and hit it. So once upon a time, Archaea and bacteria were in the kingdom Monera. And like I said, that's now debunked. I mean, well, not so much debunked as we've moved on from it. It was early, but now we split them up. Yep, in this set of notes in the chapter that you're going to read, we smash them back together again because it's a little easier. So let's do some basic information about these guys. They're prokaryotes, so they're very small, no membrane-bound nucleus or organelles. They're single-celled. unless they're all hanging out together in that microbial mat of gumminess. The monarans are further classified by their nutritional class and their reactivity with oxygen. Like I said earlier in one of the things, there are, we used to have the thought that life came because, life existed because of oxygen. As long as there's oxygen, life would go, the abiogenesis. And in the corner, they were all giggling. The bacteria and the archaea were sitting there going, oxygen, what's that? Anyway, we don't need no stinking oxygen, and it's true. So instead of phyla or divisions, they're often classified into strains, especially with infectious. So if you hit there and talk about, hear some doctors talking about a certain strain or whatnot, that's basically how we classify these guys. And bacteria can either be heterotrophs or autotrophs, which can be photoautotrophs or chemoautotrophs, which is the cyanobacteria. And we'll get into them because they spin off into their own fun mess. Where, it's kind of interesting. So anyway, remember, heterotroph means that you have to find food outside of yourself to ingest and then break it down to turn into ATP. Whereas autotrophs make their own food, usually using the photoautotrophs use the sun. And if you watch that one video I just talked about, then taste the sun. It's kind of fun. And the chemoautotrophs, which usually drink chemicals, some of the best known ones live around ocean vents, because usually what's coming out of those ocean vents is hot earth soup that's just nothing but pure minerals being spouted from the mantle up into the crust at the bottom of the ocean, and a whole, colony of life hangs out there, and we get all the fun, like the yeti crabs and the football fish and all the fun guys. So anyway, the chemoautotrophs and the cyanobacteria kind of put these two together. We'll talk about him in a minute because he's special. So how do these guys react to oxygen? Like I said, there's three flavors of this. We've got the obligate anaerobes. They were required for oxygen respiration, so they need oxygen, kind of like us. If they don't have oxygen, they die. The obligate anaerobes, they have to avoid oxygen, and they will die in its presence. So oxygen is poisonous to these guys. They can't handle it, they can't use it, and they're just like sitting there going, why? Think of the Wicked Witch of the West getting hit with water and going, what a world, except it's oxygen. So now there's the cool guys, and these are the switcheroos. These are the facultative anaerobes, and these guys are the facultative anaerobes, excuse me. These guys can use oxygen when it's available, but they can also live without it. And these guys can get everywhere. We've found them even though we build everything in clean rooms at NASA. When we shoot them up to space and back, these guys are hanging on going, that was fun. So we've found these guys everywhere. They are some of the most versatile, and therefore they don't mind where they live. You'll also find these guys as extremophobes, which are not phobes, but extremophiles. They love extreme things. We'll talk about them in a minute. So those are the three flavors of how they react to oxygen. They have to live without it, can't live without it. They can't live with it, and the swingers of the bunch going, yeah, I don't mind. I can do either, yeah. Now, again, let's go ahead and we're going to look at some of the parts of a bacterium. I know you've probably seen this before when you look at, stuff like this. But this is going to come into being some important parts of a prokaryote going forward, especially if you ever plan on taking microbiology. This is going to come back up again. So again, you will notice that they have a cell membrane, but they're usually covered in a protein coat of some sort or a cell envelope. It consists of a capsule, a cell wall. Now, their cell wall is completely different from a plant cell wall, so please keep that in mind. These guys were not even anywhere near plants yet, so completely different. So they have no membrane-bound organelles. Their DNA is usually wadded up like somebody's dirty laundry in the corner, and I do mean that. They also have ribosomes all over the place, so they have easy access straight to the DNA. So they have that cell membrane, the cell wall, again, completely different from a plant cell wall, and a capsule. And they sometimes have this in different arrangements of layers, which we're going to get into in a minute. That's really important for diagnosing these guys, especially in a medical setting. Now, they also have bacterial shapes. They come in spherical, which is the cosi or cosis, the bacilli, which are rod-shaped, the vibrio, which usually the ones like this one has a little tail on one end, and the spirillum, which is basically curly-shaped. Usually the spirillum is several living together in that shape, but they come in different shapes, and that's usually, especially, they're named like this in their scientific name. Usually it's, certain names in their scientific name tell you what group they're in according to their shape. And their shape lets them do different things as well. So that's where we're coming out with these 4 different bacterial shapes. So shape lets them do different things in their native environment. Now, there's another thing I'm going to talk about real quick. As I mentioned earlier, when it comes to the cell membrane, their cell wall, and the capsules, they can have different variations of this. And this is something we use, and you're going to learn more about this if you take microbiology, is the difference between gram-positive and gram-negative bacteria and how they layer their cell membranes and their cell walls. So if you take a gram-positive, they have this huge peptoglycan cell wall. Again, their cell wall is made of peptoglycan. Then they have a cell membrane underneath. Normal, again, what's one of the key aspects of, is it a cell? Does it have a cell membrane? If yes, it's a cell. Again, viruses don't have cell membranes. So again, once again, one more knock on them, they're not cells. They're not alive. So, because they don't have a cell membrane, they just have a protein coat. Like I said, they're just, 3 pieces of bacteria, or 3 pieces of DNA in a protein coat. Anyway, so again, and that huge peptoglycan layer right here is really, really thick. It can also be multi-layered, but I'm not going to get into that too crazy. But one of the major things is this, these guys are very weak to antibiotics, because the antibiotics can go right through this and hit the cell membrane. Even though that's really thick, it is, very low in lipid content. And usually if it's a gram-positive and you test for it, and you'll learn about that test if you haven't already done it in other classes, the gram-positive versus the gram staining. So if it's gram-positive, we can deal with it with antibiotics. These guys are susceptible to antibiotics. Good for them. Or good for us, actually. Bad for them. However, these guys with the multi-layers, which are the gram-negatives, they have a double-layered wavy cell wall. So they've got one cell wall, then they've got a thin peptoglycan layer, and then another cell membrane. So we've got 1 cell membrane, 2 cell membrane, and a very thin peptoglycan. But because there's 2 cell membranes, it's like a sandwich. This is like, when you make a bit of toast and you put a whole bunch of peanut butter on top, except in this case, you put the peanut butter in between the toast. Anyway, I like toasting my peanut butter and jelly sandwiches. I don't know about you. It's good that way, especially on a cold day. Anyway, but same thing here. These guys are, because they got this double, phospholipid bilayers and a very thin peptoglycan layer, these guys are actually highly resistant to antibiotics. So again, the reason I'm bringing this up very quickly is that keep in mind, I have a simple picture for you right here of bacteria, but keep in mind their cell membranes and their cell walls can be layered in different ways, which makes them either easy for us to kill or hard for us to kill, depending on what's going on. So gram-positives, yes, we can take care of them. Gram-negatives, that's going to be rough. They're very resistant to antibiotics, which can lead to insane resistances down the road. So if you have a doctor come in and you have a bacterial infection and he goes, it's gram-positive, here's some antibiotics, no worries, we can take care of it. If they come in and say it's gram-negative, you're going to have a little bit of a talk and a fight with these guys. So just saying, I just want you to know, I want to bring that up, that yes, this is some of their layers. So remember, bacteria are like onions. They have layers, which are like ogres, and they make people cry. Wait. Anyway. So let's go ahead and jump into archaeobacteria. So again, here's our first bunch. These guys are extremophiles. They love to live in strange places because they used to live on primitive Earth, which was a strange place. Like if we all got into Miss Royal's magic school bus that I don't own, but would be purple, and we went back in time and we got out of the magic school bus, we'd all die. Because Earth, primitive Earth, didn't have enough oxygen, was insanely hot. The floor was lava. The air was deadly. And so was the sun. So it was not a fun place to be. But archaeobacteria did not care. They developed the abilities to live in strange places, like insanely hot, acidic pools. And they still are there today. All over the place, living in strange places. Like, for instance, I have a whole bunch of their preferred conditions. We got the acidophiles. They'll live in a pH of 3 or below. So the more acidic it is, the more they're like, ah. But then you've got the reverse of that, the alkali-philes, which they live in insanely basic conditions. And you might like, basic is safer, right? No. Anyway. Thermophiles. These guys love living in insane temperatures that would boil our skin off. And then on top of that, there's the hyperthermophiles, which live in even hotter areas. Our favorite are the extremophiles living in our national parks, especially if you go out to Yellowstone. We have all of our beautiful, acid ponds. Or as we call them, hot springs, but they're acid ponds. Please do not jump in. Actually, several people have died from jumping in. And I wish I was making that up. But there are signs, literally, that says, you may look, no touchy. And then there's people that go, yay, I jump in, and then we can't get their body back because it's insanely hot, acidic pools, and the body dissolves. Fun. Anyway, look it up sometime. There have been people that have jumped in just because, or fallen in, unfortunately. There was 2 guys that unfortunately died because they let their dog off the leash, and the dog, being a water-loving dog, poor puppy, ran into the freaking pool, started screaming. The best friend of the guy who owned the dog ran in, tried to save it. Did not end well. Anyway. So psychophiles, these guys live in colder temperatures. Haliophiles love salt. They're only found in the Great Salt Lake in Utah or in the Dead Sea. So they're the only ones sitting there going, salty, bring it on. And then osmophiles, they love insanely high sugar concentrations. We use these guys sometimes. Actually, by the mean, I mean sometimes I mean the alcohol times. We'll get to that later. And thermoacidophiles, they love hot, acidic environments. The colors in this is not from the water. It's actually from the bacteria numbing on the minerals inside. So that's why they get these gorgeous variations of color in these hot springs and whatnot. So please, 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 look, no touchy. Do not jump in. Thank you. But they get these gorgeous layers of color, and that's from the bacteria. So that's literally what you're looking at with those layers and layers of color is actually the bacteria doing their job, eating all the minerals in the rocks that the acid's pulling out, and they're nom. So these are a whole bunch of happy archaeobacteria just hanging out going, yeah, we like it here. Now, Archaea comes in about 4 phyla. And some of these guys we can cultivate, and some of these guys we can't. And we're going to get into this. It's the same problem with bacteria. Archaea and bacteria have similar issues in the fact that some of these guys we can cultivate in the lab and learn more about them, and some of these we can't figure out how to make them happy and live in a lab. We can find them out in the wild, bring them back to a lab, and then they'll die. And that is an issue, trying to study these guys. So we'll get into that in a minute. But the 4 phylum are Eukaryotea, which is basically the methogens. So these guys like to live in our gut a lot of the times, and it causes flatulence in humans and other animals like cows. So you have Archaea right now living in your gut, which is totally fine. They're there to help you break down things that we can't. So they give and receive, and we get to fart. Wait. Anyway, halobacteria, this is salt-loving. They appear reddish due to the presence of bacteria's dophin in the methane, basically related to renal pigment and rhodopsin. We actually use some of these, we grow some of these to make pigments for our food. So that way we have natural pigments that we've made from just using bacteria and archaea, which is actually kind of interesting. We have this next bunch. They love sulfur. So that's Crenarchaea aota. So anyway, I don't always say these words a lot, so my bad. So anyway, these guys are very important in carbon fixation. So this group is basically sulfur-dependent extremophiles, but some of them love heat and some of them like superheat. So these are the ones you'll meet outside, and like if you ever visited Yellowstone, these are the guys hanging out and they're going hi, and you're like hello, and if they do say hi, somebody spiked your drink. Nanoarchaeota, so this basically contains one species, and that's him. Isn't he cute? They come from the bottom of the Atlantic Ocean or from a hydrothermal vent in Yellowstone National Park. That's it. That's all they come from. So isn't it nice to have your own phyla, I guess? Anyway, then we got Archaeota. So this basically is one of the most primitive forms of life. We've only found them in the hot spring of the Obsidian Pool in Yellowstone National Park. This is what they look like. That's it. Again, we can't cultivate these guys whatsoever. All these guys here, we can bring them to a lab and make them happy. These guys refuse. So they're the ultimate free people. They're like, no, you can't take me to a lab and cultivate me. So, but they look like a whole bunch of squiggly nails. But hey, who am I to judge? They probably think we're weird. So again, at some point there was some universal ancestor. What it was, we're still figuring out. And there's a lot of arguments on it. But again, it made, split out into domain bacteria, domain archaea, which we just talked about, and archaea gave rise or is related to eukaryota, which is kind of an interesting thing. So bacteria is kind of on the far side going, what are you guys doing? And they're like, inventing organelles. And bacteria is like, freaks. So anyway, so that's basically what we've got right now. Remember, further evidence might switch this up in the future, or split it out even more. Depends on how it's going. So anyway, domain bacteria is where we're going to head into next. Boop. So again, protobacteria in turn subdivided into several classes from alpha to epsilon protobacteria. I am not going to beat you with this. I'm just letting you know. These guys are basically where we got mitochondria and possibly chloroplasts came from protobacteria. So keep in mind, archaea is a different domain than bacteria. And even then, bacteria has a protobacteria right here that kind of gave rise to the rest of the bacteria. So, and the protobacteria are kind of named after basically alpha, beta, gamma, delta, epsilon, and zeta. They didn't get the funky names. We just decided to throw in the Greek letters because. Kind of sounds cool, though. What are you? I'm an epsilon protobacteria. I don't know. Just sounds neat. So anyway, now, these guys, we think, is where our mitochondria popped out of, because they actually have a lot of similarities with these guys. So we think, at one point, a eukaryotic cell rolled up to a protobacteria and said, live inside me, I'll give you all the glucose and oxygen you can shake a stick at, and if you give me some of that sweet, sweet ATP, we can hang out. And the protobacteria went, cool, and then came on in and became basically the mitochondria. That's why mitochondria has its own DNA, and we get our mitochondria from our mothers. So, if you didn't know that, thank your mom next time for your mitochondria. I say that, and then my mom just gives me the side eye and be like, stop, because I've thanked her for her mitochondria many times. Anyway, so we think they came out of the alpha bacteria. Now, while the eukaryotic chloroplasts are actually derived from cyanobacteria, which we're not even there yet. So mitochondria probably came from alpha protobacteria. Chloroplasts came from cyanobacteria because we had to have somebody figure out how to use sun energy to turn it into sugar. We weren't there yet. So if you're wondering what came first, mitochondria or chloroplasts, it was the mitochondria. Because we had things that we could steal and turn into ATP at the point. That's why everybody was eating each other. It wasn't until somebody figured out, I don't have to eat other people. I could eat the sun. Anyway, and that took a minute. Now, let's flop over to bacteria. Again, we split these into two major groups, the gram-positive and the gram-negative, that I just talked about earlier, based on their reaction to gram staining and the different layers they have of their cell membranes and cell wall. And note that all gram-positive bacteria belong to one phylum, and in the other are gram-negatives. So, notice all the gram-positives are right here, but these three right here, are all gram-negative. So, gram-negative is, unfortunately, the majority of these guys, but thankfully, they don't care about us all that much. And some of these guys we're good with. So again, the chlamydias, unfortunately, is not our friend at all. He's an obligate interracial parasite of animal cells. He is a sexually transmitted disease that can lead to blindness and going cray-cray, which is actually what happened to Al Capone. He had chlamydia, and it wasn't entirely treated, and he started losing his mind in jail. And there you go. That was a thrilling story. Anyway, spirochetes. These guys are free-living. Some are pathogenic. They have flagella. Some of these guys, again, gram-negative. So, for instance, one is the agent of syphilis. Again, another one is Lyme disease. Syphilis was also an STD. Lyme disease, you can get that from being bitten by ticks. And it's not fun. Neither of these are fun. So, again, spirochetes, not exactly our friends. Cyanobacteria, thankfully, does not care about us. Cyanobacteria, again, is what we think turned into eventually the chloroplasts that now are in cells of the plant kingdom, but also known as blue-green algae. And they are some of the first that figured out, hey, we can use sun energy and turn that into sugar. And it wasn't the best, chemical thing, but it was good. And until they, turned the planet into snowballs a couple times. We'll get into that when I start complaining about the plant kingdom. They're found in terrestrial, marine, freshwater environments. We think these guys, like I said, chloroplasts came out of these guys. And they're kind of interesting in the fact that we believe they're the most abundant photosynthetic organism on Earth, and they're pretty much responsible for generating half of the world's oxygen. So when everybody's like, no, if we don't have any trees, we won't have oxygen, and the cyanobacteria are just standing there with their little flagella on their non-existent hips going, excuse you, because they're the ones making up most of your oxygen. So thank your cyanobacteria. If you've ever played with algae in a pond, you probably poke these guys and they're like, dude. Then, last but not least, is the gram-positive. So these are the ones we can kill with antibiotics. These usually are soil-dwelling members. They decompose organic matter. Some cause disease. They have a thick cell wall, and they do not have that outer secondary membrane like the gram-negatives do. So again, we've got the ones that cause anthrax, the one that cause botulism, causes diarrhea during antibiotic therapy. That's fun. But they're also the ones that we really like to smell. I'm not kidding you in the fact that humans actually have a weird built-in sensor for good earth. In other words, good fertile ground for growing crops and whatnot over the generations. It's why some people really like sniffing dirt. And I don't mean like that. I mean, have you ever gone out? Who's a gardener? Are you guys garden? Have you ever gone out in the garden and you've turned some soil and you're like, and that smell that comes up off of rich, fertile soil? Humans are actually detectors of the good bacteria inside of soil, it's been shown, because we were so, we still are very dependent on agriculture. So it's actually kind of interesting that's why some people, especially if you're a gardener and whatnot, you've probably noticed that smell, not with the fertilizer of poo, but, good dirt smell that makes you go, that actually has a name. I'll put it up in post here. But anyway, so some of these guys, like I said, actually are really good, and we love using them, and we make sure they're still alive. And some of these also live in our gut and help us break down food and whatnot. So bacteria, it can be good, it can be bad. And again, here is the positive or negative. Again, what we've got with the positive, if you got that really thick outer wall, and then and then it's cell membrane. Again, we can it stains purple violet after a gram stain. It's easily treatable with antibiotics. Gram-negative, on the other hand, this is what gram-negative looks like after a gram stain. It's usually red-pink. What happens normally is we take this violet and we stain both of them, but then after a while, and they both turn purple, but then we wash it off. And because these guys have a lot of that really thick wall, they stay purple, where these guys go colorless again, and then we stain them with a red or a pink stain afterwards, and they'll turn pink, and these guys will stay purple. And that's how we can figure out who's gram-positive and who's gram-negative. Like I said, this is a very famous lab. You may, if you've already taken micro, you've probably done it, or some people I know have done it in high school too, depending on who your teacher was. So anyway, these guys are harder to treat. With antibiotics, they're very resistant to it. These guys are easy to kill with antibiotics. So, again, that's why we split them into two. And, yay. Now, how do these guys reproduce? And we already kind of went over that back in 111, and that's binary fission. But it makes two identical clones. So how does bacteria exchange DNA? Which is kind of interesting. So again, one of the things is called transformation. And bacteria and archaea love to kind of play our version of Pokemon cards. When you're younger, or maybe not, or maybe it was Yu-Gi-Oh cards, or whatever. Basically, or baseball cards for way back when. Basically what happens is these DNA usually is put into these loops. They're called plasmids, and this is actually how we make bacteria make things like insulin for us and whatnot. And if you put in DNA into these loops and you just put them free floating around with some bacteria just hanging out doing their thing, they're actually attracted to it. They'll float on over and go, "Oh, I don't have that plasmid," or "Oh, look at the shiny Pikachu card," or that shiny Charizard card, and they grab it and they stick it in their collection, except their collection is their whole DNA. So they do that, and they actually will bump into each other on occasion and literally basically swap DNA, and that's conjugation. So they'll bump into each other, they'll make this bridge between each other, and literally one will hand out plasmids to the other, and vice versa, basically sitting there going, "Oh, I've got this, do you have this?" And they trade. So they'll trade plasmids through conjugation. So it's kind of interesting. So one is, we actually take advantage of this and having them pick up DNA, for instance, to make human insulin. So we put it in a plasmid, we cut it out, put it in a plasmid form, it goes in, they absorb it, and then all of a sudden they start, as long as we keep them alive and happy, and they'll start pumping out insulin, we purify it, and then we give it to diabetics. That's literally how we make human insulin with bacteria via transformation, because these guys just have going, "Oh, I don't have that DNA." They've got a FOMO, fear of missing out on the DNA plasmid. They have that. The other way is transduction, and that is using a virus to inject DNA or RNA in them, which is interesting because we have used that again against bacteria. But we've also, like I said, our buddy the bacteriophage loves going over just bacteria. And if you watch that video of the ultimate and most ancient war on Earth between bacteria and viruses, that's basically what's going on right here. So sometimes we are, using viruses to actually inject. This you might be like, but why? Because this. And the reason is these guys don't always get picked up. Sometimes some of these pick up, and some guys, in the corner is like, I don't want it. No. So, so one kid in the corner is just like, no, I don't want to eat that. So, but with this, we can get them injected every single time using viruses. So in the future, we might be using all our bacteriophages to do things to our bacteria. Because thankfully, bacteriophage viruses only like bacteria and nothing else. And I hope it stays that way. Now, culturing these guys is complex, and one of the greatest discoveries was by a German physician named Robert Koch, and it basically is credited with discovering the techniques for pure culture, which is straining and using growth media. Microbiologists basically grow prokaryotes in the laboratory using culture mediums containing all the nutrients that they want. The medium can be liquid, broth, or solid. Again, if you take microbiology, you'll be using all of these types usually. And the incubation time is usually you got the right temperature. That's why we always have these incubation ovens in the back of a lab and whatnot. And this should be evidence of, microbial growth. And Koch's assistant, Julius Petri, invented the Petri dish, which is, if you haven't heard of a Petri dish, then I don't know what to tell you. But anyway, so we do culture quite a few, but over 99% of bacteria and archaea are unculturable. This is due to a lack of knowledge, because we can't always sift everything out that they need from an environment, what to feed them and how to grow them, and they might have wacky, requirements for growth that we just haven't observed, and it's really hard to observe. You can't really always take a electron microscope outside. You can't. And stare at things. So that's why we have, things that cannot be cultured but are not dead or in a viable non-culturable state, or VBNC. And this occurs when the prokaryotes respond to environmental stressors, and they'll go into a dormant state. It's kind of like protecting itself. So they'll basically harden and protect themselves until the environment becomes better for them to survive. And this is how a lot of archaea and bacteria have survived over the millennia, is literally if something's going wrong and the environment's bad, they'll literally harden, and then when the environment gets better, they'll loosen up and go back to doing what they did, because that dormant state allows them to survive pretty much anything, including going into the depths of space. So, because we found them on our spaceships that have hitched rides back and forth and scraped them off, and then when they hit the environment they like, they're like, oh, that was weird. So anyway, now it's called a process called resuscitation, and basically the prokaryote can go back to normal life when environmental conditions improve. And this has allowed them to survive the test of life getting wiped several times on this planet. And we're going to talk about that in a future lecture as well, how many times life has gotten nearly completely wiped off this planet, which is 5, and we're in the middle of the 6th. So we'll talk more about that later. So again, how do they get their food? We got the autotrophs and the heterotrophs. The autotrophs, basically, they make their own food, usually either photoautotroph, which is light, or chemoautotrophs, which is, guys that usually, especially the sulfur-based ones will do this. They use inorganic chemicals. Normally, they're found around ocean vents on the bottom of the ocean that's venting a whole bunch of inorganic chemicals, or hot earth soup, yum yum. Heterotrophs are either photoheterotrophs, which means they can kind of flip back and forth between, if they've got light, cool, they can make their own food. If not, they'll go eat Steve. Whoever Steve is. And then the chemoheterotroph, which is kind of like us. We're protist fungi plants, and some plants and animals. We're chemoheterotrophs. In other words, we have to ingest something to break down, and why we have a digestive tract. So the chemoheterotroph went on and became the prokaryotic kingdom, the fungi kingdom, and the animal kingdom, whereas the photoautotroph went on to become plants and algae and some protists. So those were the two wieners. Not that I'm saying the other two aren't wieners. I'm just saying they're not as everywhere. So again, these guys are insanely important. We're going to talk about this again, But the carbon cycle and the nitrogen cycle, if it wasn't for bacteria, we would not have this. And even the plants need this. We can't pull carbon and nitrogen from its gaseous form into our bodies. It doesn't work. Our atmosphere is mostly nitrogen, and nitrogen is something we need to make our proteins in our body. But we can't breathe in nitrogen because we can't do anything with nitrogen into because it's kind of inert. It doesn't really do anything, and therefore our body's like, I can't do anything with this. It's inert. I can't react. Unlike oxygen, which is very reactive, that's why we can use oxygen. It's very reactive. Nitrogen, not so much. You can poke nitrogen. Nitrogen's like, what? So we can't do anything with it, but we do have nitrogen-fixing bacteria in the soil, and working with plants and fungi, these guys can turn it basically into nitrates and nitrites. And nitrates and nitrites can then be turned and used by plants, fungi, and animals, especially animals eating plants and fungi. Although then when we die, the fungi eats us. It's the great circle of fungi. Anyway, or nitrogen in this case. So if it wasn't for plants with bacteria and fungi, we wouldn't have nitrogen to make our own proteins so we can get big in bulk. Get them gains. Same thing with the carbon cycle. Basically, microbial respiration and decomposition, so we can take it from the fossil carbon and also the carbon going around. We need to keep that carbon cycle going because if we have too much carbon dioxide in the atmosphere, it gets way too hot. Kind of now. So anyway, it's a long story. But anyway, so we got to keep the carbon moving as well, so that way it can go in and out of living things just as much as the nitrogen goes in and out of living things. So without carbon and without nitrogen, we really wouldn't be very large. So thank you, bacteria, for fixing our carbon and nitrogen for us. All right. Also, there's other things. Let's talk about how good they can be. We like prokaryotes. They interact with us in a number of ways. Like I said, the carbon and nitrogen cycles, huge. They also produce nutrients on our digestive tracts, so they live in our digestive tracts, and we're fine with that because, they help us digest things and get things back that we wouldn't necessarily get from our own food. Granted, byproduct is farting, but hey, it's a give and take. Without these guys, we would not have cheese, bread, wine, beer, or yogurt. So if you like any of those 5 things, thank you, bacteria. Microbial bioremediation is basically using prokaryotes to remove pollutants. We've actually developed some bacteria that will eat oil spills. So if we have an oil spill, oil tanker spills, we can put these guys out and they'll go nom 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 nom, and then they die off when they run out of oil to eat. So that's fun. And again, I can't say this enough, nitrogen fixation. We eukaryotes cannot pull nitrogen from the atmosphere, so they give it to us in the form of ammonia. And if you've ever sniffed ammonia and all your nose hairs fall out, don't do that. But anyway, still, we need that. Not straight though. Anyway, so yay, prokaryotes, we love them, for we love cheese, bread, wine, beer, and yogurt. However, there's a lot of them that see us as dinner, and not in a good way. So remember, viruses and bacteria are not the same thing. Viruses are very tiny. They're not a living thing. Bacteria are a living thing. However, bacteria has caused many, 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 many infectious diseases. So as back as 3000 BC, we have had many, and unfortunately, a lot of memorable pandemics that led to the decline of pretty much, and had to rework, almost killed off the whole human population. The plague of Athens was done by salmonella, which also is typhoid fever, which also led to typhoid fever. Yum! So remember, wash your food. I guess they weren't. Bubonic plague, Black Death, that's another one. It's Yersinia pestis. He was a bacteria, not a virus. Zoonoses. So these are basically, they infect animals but can be transmitted to humans. Unfortunately, necrotizing flesh-eating bacteria is one of them. So again, civilization as it arose from humans, as I talked back in the viruses thing, yeah, bacteria can do that jump too, a little bit better than viruses can. So that's where flesh-eating bacteria came from. Yay. So, characteristics of foodborne illnesses have changed over time. In the past, it was common to hear about, different cases of botulism, because there's a fatal disease produced by a toxin from the anaerobic bacterium Clostridium botulism, but it's hysterical because now we don't really hear about it, and we've actually taken that bacteria, sucked out its paralyzing agent, and turned it into Botox. I'm not kidding you. Yes, the bacteria that causes botulism, we culture it in a lab, suck out its toxins, purify it, and then inject it into our muscles to freeze our muscles so we don't get wrinkles. Yeah, we did that. Anyway, Staphylococcus aureus, also known as staph, is common, can live in the human body. It's actually living on our skin right now, unfortunately. And that's where staph infections can come from. You get a cut, it isn't kept clean, the staph can get in there. But we literally have staph kind of living on our skin right now. And now you're probably going to be like, I'm going to go take a shower. But it's usually easily treated with antibiotics. However, a dangerous strain has arisen because we overused antibiotics. We overused the thing that can knock bacteria back down again and made it so we can survive, which is the invention of penicillin and other things like it, like methicillin. So unfortunately, that's where MRSA is. It's methicillin-resistant Staphylococcus aureus, or MRSA, which is not good. If you've got MRSA, I hope you never get MRSA. I hope nobody gets MRSA. But anyway, so that's our buddies. Now, if you watch Hank here, he smashes Archaea bacteria and Protista in the one go. I'm going to split Protista into the next lecture, and that's the next one we're going to be coming up on. So, I think that's the last...
We've spent the past...
Yeah, that's the last... And then we've got the Amoeba Sisters. She talks about the nitrogen fixing in here, which is extremely important. No. All right, but anyway. And that's it for Archaea and bacteria. And now we're going to move on to the next batch, which is the prokaryotes, or as I like to call it, Kingdom Protista, which is like the junk drawer of the kingdoms of this earth. And I'll get into exactly why it's a junk drawer in the next lecture. So until then, have a good one.